Hello everybody, this is the TMS demo for 3031, and I'm going to show you just the TMS machine over here. Isn't that nice? Right, so having a little bit of an issue at the moment because I'd like to show you, turn it on, how it works and how it looks like when it's ready and everything. But the problem is that my postdoc has the power cord, so we keep that in a separate place so people don't just wander in here and start zapping themselves or friends or whatever. And she hasn't been here since the pandemic started, basically, so I can't actually turn it on for you. I apologize for that. But I will kind of show you a little bit of a run through and what you would see and what you might expect if it were turned on. I know that's not quite the same, but yeah, I'm afraid it's the best I can do under the circumstances. So if we could turn it on, what we would see is we have this screen here, and this is the interface for the TMS machine. It has these little buttons on it and the dial. The buttons are used to arm the machine, so green means ready to go, yellow means pause, and red means stop. And the dial is used to adjust different parameters, most usually the one related to the intensity of the stimulation. So you recall from the lecture that I talked a bit about how we look for a threshold, a motor threshold. So we will test people, start to stimulate them over their motor cortex and look for a visible response in their contralateral hand. And because some people's skulls are thicker than others, sometimes you need more intensity to get that visible response for some people than we do for others. So let's say it was me and I'm at 50% of intensity and I don't see any response, and then I turn it up to 55 maybe, and then I see a little twitch. Okay, then I would argue that, or I would make the conclusion that my motor threshold was 55%. And from that, we can use that as a baseline in order to then adjust that upwards or downwards depending on the percentage of motor threshold that we're running the experiment at. And there's all sorts of safety limits and considerations that you have to take into account. So certain number of pulses, or you're limited to a certain number of pulses at each percentage of motor threshold. So if you're testing someone at 115% of motor threshold, you're limited to a certain lower number of pulses in a 24-hour period than if you're testing someone at 85% of motor threshold, where you can give them more pulses safely. Again, safety is obviously going to be a primary concern because you don't want to go over the limit of what's a safe amount of stimulation and just start, taking the risk of giving someone a seizure. As I talked about, we also have obviously contraindications for people to be tested, so you can't have people who come in with a family history of seizures or who have any neurological conditions or who are not in a reasonable physical state, that is well rested and not currently on drugs or anything like that. So those are other considerations to take into account. Now, obviously, again, I can't show you what it would be like to stimulate me, but you can see on the videos that the one video at least that I posted before the lecture that you put the thing on the person's head and it goes tick 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 if you're doing repetitive TMS, and then they have a particular response. If it were a motor threshold test that we were doing, for example, you might have the TMS coil might go on my head, and then you might look at my finger, my thumb and finger, and see if there's a response to that, and it might just kind of look like that, just a little twitch. And it's a very strange experience if you're actually the person getting TMS because you don't mean to move, but your body moves anyways, right? So it's an involuntary reaction that you have no power over. So it's quite a strange experience, but kind of cool at the same time. What else can I tell you about? Well, we also have a means of locating a particular part of the brain that we want to stimulate. and that is using this handy dandy little TMS cap, which we got from the fine people at Magsim. And what we would do is we would put this on the participant's head. So it looked like this. Ain't that nice? I feel like I'm Amish or something now, but here you go. It's quite snug fit on my big fat head as well. But anyways, this would go on, and then you could, I think I picked the smallest cap that we have. They come in different sizes. Anyways, this goes on, and then, as I talked about in the lecture, we measure across the top of the head as well as from the front to the back of the head. And we use that to locate the vertex of the skull. So in the experiment that we're doing now, or about to start again, or in any experiment really, you would be looking for the vertex of the skull, and you would start with this rather inconveniently placed little notch at the front of the ear. And I have to take the cap off a bit to show you. So I was talking about this in the lecture, and it's actually this thing. I forget what the name is for it, but you put the tape measure, you start one end of the tape measure there, you wrap it around the head, over the top of the head, something like that, and then you hold the other one, and then you measure and you say, okay, how far is it from one ear flappy thing to the other? And half of that distance is then obviously the top, right? So the top of the head. Similarly, you start at the bridge of the nose, and you go over the top of the head, and you go back to, and I won't be able to show you this, but at the back of your head, there's a little indentation, a little kind of knobby thing there at the base of the skull, and you go from the bridge of the nose to that, and then half of that distance, then you have a cross index of halfway from front to back and side to side, and that would be considered the vertex of the skull, the very tip of the skull, basically. And once you know that, then you have the ability to take measurements based on what we understand about anatomy. So in our study, we're trying to impact or to TMS the dorsolateral prefrontal cortex. So we would move a certain number of centimeters forward from the vertex, and I believe we are doing the right hemisphere, DLPFC. So it's a certain number of centimeters forward and then a certain number of centimeters to the right. We have that location, we've measured that out, okay, from the vertex to where we want to stimulate. Then we just put a little marker there. So we have some of these that we got for, is this showing it to you? There, like that. And then we just put that where our stimulation point is. And then from then on, we know where we're going to stimulate. So as long as the cap is nice and snug, it isn't moving around on the person's head, then we just have that spot in the same place, and we know that, okay, we just go back to that spot every time and zap them there. And then the idea being that that will maintain the integrity of the experiment in as much as we're always stimulating the same parts of the brain. Okay? So that's how that all works. Don't know what else I can really tell you. I don't think I've shown you the coil yet, have I? So this is the coil and that we use. This is the figure 8 coil. Okay, so you recall from the lecture that you can have a circular coil, you can have a figure 8 coil. The figure 8 coil works better because of the way that the magnetic field is sort of generated. It comes from, it follows the circle of the current, as it will, and because we have a figure 8 current, then the main focus of the ultimate magnetic field will be in the center of these two figure 8s. And I can't really show you what I mean, but you know what the center means, right? It's right there. Right? And so that will be the area that we put over the area we want to stimulate. All right? So the machine allows the facility to do both single pulse TMS, and these are the buttons that you would use to release the current, so to speak. So you'd go click the buttons, and then jit. If it were single pulse TMS, it wouldn't go jit, it would go tick, like that, like you heard in the video. So tick, and then that's a single pulse of TMS going through my skull into my brain. And we can also adjust the dials on here that you can't see because it's not turned on, but we can also adjust the dials to make it use repetitive TMS. And in that case, we might do it at, say, 10 hertz for 500 milliseconds. So there would be tick, 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 tick. Okay? And then there would be, this would result in a longer lasting disruption than just the single pulse. Obviously, because you're limited through safety concerns to a given number of pulses in a 24-hour period, you can give more single pulses or do it for longer because you're only doing it 1 per second, dunk, 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 then you can, if you're doing on each trial, if you're only going dunk and then the next trial you go dunk again and then the next trial dunk, then you can do more, you can do that for longer than if on every trial you're doing dunk, 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 right? Because for every 1 pulse and single pulse, you'll be giving 5 in repetitive pulses, and you'll run out of safety limits in the repetitive condition more quickly than you would in the single pulse condition. Okay? Now, the thing about single pulse is obviously it only is going to disrupt that part of the brain for 50 to 100 milliseconds, so you have to be sure that whatever you're testing them on is going to be affected in that time period. So you have to time the pulse quickly or precisely to know that it's affecting whatever you're trying to measure that they're doing right after that pulse happens. But another way you can use single pulse is to simply have them sit there and administer it repetitively for a period of time. And we actually, this is what we're doing in this current experiment where we're stimulating the dorsolateral prefrontal cortex, is we first have them do various tasks. So I won't bore you with the details, but the tasks we're interested in motor imagery and overt action tasks, as well as certain cognitive kinds of tasks that look at their executive functions. And then once they do those that first time, that gives us a baseline measure of how they do those tasks. Then we have them sit down and then we stimulate them at 1 per hertz stimulation, so 1 pulse per second over the right dorsolateral prefrontal cortex, and we continue that for 30 minutes. So they just sit there and go doop, doop, doop once per second for 30 minutes. Now this gives them a total of 60 times 30, so 1,800 pulses. That's a safe limit. We also don't have it too high, so we've got a little leeway in how many pulses we can give them. It can actually be a fairly good, large number. So that's okay. But the result of this is that this continuous sort of slow stimulation has a similar to effect in the end after a half an hour as you would have if you were just giving them a lot of pulses in a hurry. Okay? But it also has the benefit that that effect then lasts for a little bit longer. So you're essentially kind of scrambling the brain a bit each time with each pulse. But after 1,800 pulses, you've scrambled it up pretty good. Okay? They can still walk and talk and chew gum and stuff, but there's going to be some effect in how their brain functions. But at the same time, you're not giving them so many pulses at once that it becomes a safety concern. So this single pulse sort of continuing over a half an hour kind of thing will then result in effects on their brain function that themselves will last for about half an hour to an hour. Okay, so then we give them the TMS. Other participants get what's called sham TMS, and effectively all this involves is turning the coil over the other way. Okay? So if this is the zapping side, right? So I'm getting zapped. Now if I flip it over this way, the orientation of the coil is such that the pulse is actually going away from my head, but it still feels the same. I hear the click and I feel a little on my scalp. Then in that instance, half of the people will get the sham TMS, so they think they're being stimulated, but they're not, and they serve as the control group. Okay, so half the people are having their brain scramble a little bit, the other half are not, even though they think they are, and then we go back and we run the same tests again, the motor imagery, the overt action, the various cognitive tasks of executive function, and we see how does the TMS affect them. Okay, so that's the current experiment we've done. Now, normally, if this were like we were all sort of here together and we could, freely cough on each other and everything, there would be the opportunity for you to interact and ask questions and, you know, hear the thing in person and everything. And so I'm sorry that we can't do that this time. Also sorry that I don't have the power to supply, so I could turn on, give myself a few good zaps for your enjoyment. But I could, simulate that what happens when I get TMS, right? But it would only be a bit